welcome to this exciting session that we have in store for you. The title of the session is Reversing Cancer with Immunotherapies and Exosomes. And before I introduce the speakers, I'm going to take just a few minutes to introduce the Nova Southeastern University Cell Therapy Institute, which I direct. And the reason, and this is part of the new, so this is a new Cell Therapy Research Institute at Nova Southeastern in the new Nova Southeastern MD Medical College, and our focus is on cancer immunotherapy and regenerative medicine. And the reason I'm putting this up here is that a lot of times people ask me, well, you know, what is the NSU Cell Therapy Institute? What do you do? Where are you? And so I just want to share with you a couple slides. We're at the Center for Collaborative Research at Nova Southeastern in Fort Lauderdale. This is the building we're in, the CCR. It's one of Florida's newest and most advanced research buildings. It's over 200,000 square feet, over 6 floors. It houses not only the NSU Cell Therapy Institute, but several other research institutes and ranging a wide variety of topics. And also want to mention that we have a very strong collaboration with leading medical scientists from Sweden's Karolinska Institute in Stockholm. And you'll be hearing from one of our distinguished Karolinska colleagues in just a moment. This is my last slide. I just want to mention that the Center for Collaborative Research has state-of-the-art shared core facilities, and we share it not only amongst ourselves in the center, but also this is available to other academic institutions in Florida and elsewhere, as well as industry partners. So we welcome anyone in the audience who is interested in using these facilities. Include flow cytometry. This is state-of-the-art flow cytometer for sorting. Genomics core, all state-of-the-art again. We have a cell therapies core, which is an interesting and perhaps a little different from what many of you may be used to. But this is a clean room for process development. And so to go from the laboratory experiment to CGMP, it's very important to do the scale up and characterization and so forth. So we have that ability to do that, and for both stem cells and immune cells, and also an imaging core with a again, state-of-the-art confocal microscope, and we're adding a electron microscope very shortly. So again, this is available to anyone who is interested in using these facilities. Just go to our website, you go to NSU, and you will find more information on the Center for Collaborative Research. So with that, I'd like to introduce our first speaker, our distinguished guest from Karolinska Institutet, Dr. Hans-Gustaf Ljunggren. He's going to talk on the topic of NK cell therapy for cancer. I just want to say a few words about Hans-Gustaf. I've known him for many years. He's the former Dean of Research at the Karolinska Institutet. He's the founding director of the Infectious Diseases Research Institute and Clinic at Karolinska. And he's also been a pioneer in the natural killer of cell field in both infectious diseases as well as cancer. And today, as I mentioned, he's going to talk about the role of NK cells in cancer immunotherapy. So please welcome Dr. Ljunggren to this podium.
So, thank you so much, Rich. Thank you for the invitation, and I should say it's really a pleasure to be here at this meeting. There have certainly been many many meetings in the meeting for many of us. Really a fantastic place to be at. And I shall also say that we, of course, appreciate our interactions and collaborations with the scientists at Nova Southeastern very much. So I will give you one story today from our laboratory, and that's on our attempts to use human NK cells to treat malignancies in patients. So I think I press here to... Now it works, okay. Some disclosures that does not really relate to the present presentation. So this was a project that started almost some 10 years ago. And it was a field that was completely new to us going into clinical therapies. We were basic scientists, translational scientists, but really didn't have any experience in clinical cell therapy. But we had been working on NK cells for a long time. We've been characterizing their abilities to kill tumor cells in vitro, in mouse models, ex vivo in human settings. About 10 years ago, we started to contemplate as to whether it shouldn't be possible to use these cells also in settings of immunotherapy to combat human malignancies. So to educate ourselves, we tried to summarize all the available knowledge on the topic in this particular paper published 2007. And I think I can almost read from here that we said that current insights into molecular specificities that regulate natural killer cells suggest, and that was really what we thought at the time, that it might be possible to design NK cell-based immunotherapy strategies against human cancer. And as a rationale for this, we did go through all the evidence for NK cell interaction with human tumors, and in particular, all the aspects that one had to think about at that time with the knowledge that they had then in order to advance NK cells into the clinic. And we ended up by sort of predicting that if we would use NK cells in the right way, they should either alone or in combination with other therapies really have a future in treatment of human cancer. So that was sort of the starting point. In the review, we addressed a number of different things that were really open at that time. One of the questions was, of course, what type of patients would one go for? A big question at the time was whether one should try primarily to target solid cancers, or should one rather go for liquid cancers, hematological malignancies. And of course, if we would go, let's say, for hematological malignancies, second question becomes sort of what type of hematological malignancies would you go for? Lymphoid, myeloid, or different subgroups they're under. We also thought a lot about the conditions. Would we be limited towards incurable conditions, or would there even be settings of minimal residual disease that we could use these cells under? And then at the time we'd been working, as I said, a lot with ex vivo studies of natural killer cells being able and their ability to target human cancers. So we wondered at the time whether it would be necessary or whether it would be important to pre-test the patient's own malignant cells against killing by natural killer cells to form a rationale for going into the patient or not. We also speculated as to whether it would be of value to measure MHC class I expression on the patient's tumor cells, as class I deficient tumors would possibly be more easily targeted than class I expressing ones, at least under certain circumstances. Another thing that we thought quite a lot about was how to, or was the source of NK cells to be used if one wanted to use NK cells in settings of clinical trials. Peripheral blood-derived NK cells was, of course, an option, but one could also envisage that one would try to derive NK cells either from cord blood stem cells or from bone marrow hematopoietic stem cells. And at the time, the IPS technology has just been described, so thoughts were also developed along the line of trying to generate iPS cell-derived NK cells. And in parallel with that, there are also NK cell leukemias, some of them with retained cytotoxicity, and others are trying to explore those in settings of immunotherapy. They, of course, have some limits in the sense that they are tumor cells themselves, so they have to be radiated in order not to proliferate in the patient, and because of that, they will not proliferate. So there are some limitations there. We discussed mostly around the use of peripheral blood NK cells, and one big question was, of course, if we would go for peripheral blood NK cells, would we use autologous or allogeneic donors? And furthermore, if we would use allogeneic donors, would it be so that certain donors would be better, more suitable than others, for example, related donors? Next question sort of came about was how to isolate and prepare NK cells from selected donors. Would we, for example, go for peripheral blood-derived NK cells? How would we collect them? Would we use harvested cells directly as a bulk population, or would we deplete them, the PBMCs, from a number of different cellular subpopulations other than NK cells, or would we even positively enrich NK cells from these populations? Next question was as to whether one should activate or expand NK cells ex vivo prior to infusion. And with that comes a number of questions relating to the choice of media. It has to be GMP approved, cytokines, other molecules, possible feeders to support survival of the NK cells, activation and proliferation, and of course the culture conditions itself, depending on how long we would like to culture the cells. If we would go for allogeneic cells, we obviously have to condition the patients in order to avoid reaction of the cells that we inoculate into the patients. And there were some different strategies even there. For T-cell therapy, Steve Rosenberg had described a high-intensity myeloablative conditioning strategy that would have some advantages even for us, because it would likely allow maximal expansion of NK cells in the patient. It would quite efficiently prevent reaction of donor NK cells. It would most likely eradicate regulatory T cells that also suppress NK cell activity. And of course, with the depletion of a number of cells, it would reduce competition for NK cell growth factors, including IL-15. But the drawback is, of course, that this protocol would have or could have severe side effects that may pose some clinical challenges. So the alternative, we speculated, would be, after all, a reduced intensity conditioning that would avoid at least some of these side effects, but at the same time would not be associated, or that would have some of the beneficial effects, but at the same time would not have all the side effects that a high-intensity conditioning would have. And then, of course, comes questions relating to the study design, single, multiple doses, supportive treatment, IL-2, etc., all things that we contemplated about. Finally, we sort of decided to go to patients with hematological malignancies, and in particular patients with primary chemotherapy refractory or relapsed high-risk myelodysplastic syndromes, or patients with secondary AML, or patients with de novo AML, all of them not being eligible for hematopoietic stem cell transplantation due to tumor burden and resistance to chemotherapy. Rather wide age group we opted for. We did want the patients to have a certain grade of clinical performance state. They should not be immediately dying. at least an estimated survival time of 3 months. We opted for, because in part it was the easiest choice, to try to generate a peripheral blood-derived NK cell enriched product obtained from selected, very well characterized haploidentical donors, and there's a specific rationale for that I will not go into, but it has to do with a KIR-KIR ligand mismatching that we wanted to achieve. Decided that we would isolate cells via leukapheresis process and that we would deplete the products extensively from contaminating or from T cells and B cells. We decided at the time, we would probably have decided differently now, but not to expand the cells in vivo, just to overnight activate them with IL-2. One of the ideas at the time was that an extensively expanded population would maybe have lesser of an ability to further expand in the patients, and we wanted to give the patient an optimal chance to expand in the patient. And we also set up a number of standard quality control or release criteria. We opted for reduced non-myeloablative conditioning with intermediate doses of Cy/Flu, and also instead of using total body irradiation, we decided to go for titrated doses of total lymphoid irradiation only. We opted for infusion of overnight activated haploidentical NK cells following condition with a single dose, and also we made the decision not to subsequently provide the patients with IL-2. It's difficult to say at this stage whether that was a good decision or not. We know that IL-2 would contribute towards activating Tregs that would suppress the NK cells. On the other hand, IL-2 would also support NK cells, but that was our decision, and this is the conditioning escalation schedule that we made up. As primary endpoints, we put safety as number 1, and we also added the NK cell expansion as a primary NK cell endpoint, and then as secondary endpoints, achievement on complete remission at 12 weeks following NK cell infusion. And also we opted for, and this was new, we really wanted to perform an extensive monitoring, including a detailed phenotypic and functional evaluation of the NK cells following infusion, if that would be possible. So with all this, we went through the standard process that was completely new to us, with preparation of all the essential documents and logistics. It was a period of some 2 years to do this. And in parallel, we did several rounds of validation studies in our certified GMP laboratory, Vecura, that we have at the Karolinska Institute. We also, at the time, set up all the clinical interactions that we needed, and of course, primed our physicians for the identification of patients and donors, and all the logistics with the sample flow that I have in the next slide. So in 2011, we registered the study with the following title: In vivo expansion and efficacy of adoptive natural killer cell-based immunotherapy for high-risk myeloid diseases. And this is just to show the logistics of the process. This is the flow of the cells. This is the flow of the donors. This is the patient flow and visits, and these are all the collaborative clinics and units at the Karolinska University Hospital involved in this study. So it's, as is often the case in clinical trials, quite a complicated logistic endeavor in itself. So in 2012, we were ready to go. This is my colleague, Kalle Malmberg, who has been instrumental in many parts of this trial, together with a young physician scientist, Andreas Björklund, who became our physician at site, took care of many of our patients. So the study went on over 4 years. We included about 4 patients per year, and ended 2016 at the time when we had included in total 16 patients. Turned out to be 5 patients with high-risk MDS, 8 with MDS AML, and 3 patients with de novo AML. As I mentioned, all of them were refractory to first-line chemotherapy and ineligible for hematopoietic stem cell transplantation. Many of them had been through a number of prior therapies before, towards which they had not responded or only partially responded. Some of them had undergone an allogeneic hematopoietic stem cell transplantation prior to the study and then relapsed. And the medium age or age span was between 40 and 70. All patients received lymphodepleting chemotherapy according to the study plan outlined here in the schedule here, and we just have all the details in these tables, which are probably impossible to see for you, at least for me here, but it doesn't really matter. NK cells were obtained from selected haploidentical donors, in all cases then 2, where there was no sufficient donor. So then we went for an HLA match donor. And all of this was according to study plan. And now to the clinical results. So first of all, and which is important, all NK cell infusions were well tolerated. We had no general severe non-infectious or non-NK cell related or NK cell related toxicity observed during the trial or the whole follow-up. And this is in line with other more recent NK cell trials also, where the cells are rather safe in patients. Very much to our excitement, I must say, turned out that 9 patients responded to the treatment. 6 patients achieved an objective response with complete remission, marrow complete remission, or partial remission in combination with improved hematological parameters. 3 patients achieved stable disease or a morphologic leukemia-free state. Interestingly, because of the treatment, 6 of the patients became eligible for an allogeneic hematopoietic stem cell transplant, and 5 of them proceeded to transplantation. 1 of them could not due to due to the inability of a suitable donor. And following a period of 3 years, 4 of the 5 transplanted patients are free from disease, 1 of them now for more than 5 years. We did a quite extensive immunomonitoring of patients where we tried to correlate responses or immunological correlates to patients that responded versus non-responders. And I'll just show you a few selected observations. One of the observations was that in all major responders, donor NK cells were detected at day 7 or 14 post-NK cell infusion, showed here, whereas donor NK cells were only detected in about half of the patients in minor non-responders. So possibly in these NK cells had been reacted or at least disappeared in other ways, in contrast to the responding patients. A very interesting observation for us working with NK cells were that we saw among residual tumor blasts in the responding patients signs of immunoediting. This immunoediting went completely in the opposite direction, as you would see in a T-cell immunotherapy. Patients that had responded had on the residual blast cells, higher levels of total class 1 or HLA-E shown here. So this is the non-responder population pre and post-treatment, no change in HLA class 1 total or in HLA-E expression, whereas in the responder, residual tumor cells in the responder populations shows increased levels of MHC class 1 expression and HLA-E. To me, this is the first time this has been seen in vivo signs of immunoediting by NK cells, leading actually to higher expression of HLA, which is of course totally in line with what we know about the molecular specificity of the cells. We also saw in all major responders that the patients displayed specific losses of tumor cell clones, some of the clones which carried poor prognosis mutations, telling to us that treatment with this protocol can really eradicate leukemic clones bearing high-risk mutations, and I'll not go into the data here. We also made the observation that in major responders, there was a less pronounced activation of CD8 T cells and in general lower levels of inflammatory cytokines following NK cell infusion than non-minor responders. Despite the fact that we did not give IL-2 to the patients, we did see in all patients various degrees of Treg activation. So the study was just recently accepted for publication in Clinical Cancer Research, so if anyone is interested, you can go to the journal. It will probably appear in a couple of days or weeks. So just to conclude the study, first of all, with respect to safety, all NK cell infusions were well tolerated with only a few transient adverse events. We saw an objective response with complete remission, marrow complete remission, or partial remission in 6 out of 16 patients, a minor response in 3 out of 16 patients. 5 of the patients proceeded to allogeneic stem cell transplantation, 4 of which are now free from disease for more than 3 years, and 1 of them, as I said, for more than 5 years. Evidence for this being mediated, the responses being mediated by NK cells, I think or correlates at least with that, is the fact that NK cell expansion in vivo correlated very much with clinical responses, and also the observations on immunoselection or on immunoediting on remaining residual tumor cells in the patients. So the implications of these findings are that this is a protocol, and it can, of course, and will be refined in the future, be a protocol that can induce major responses in patients with MDS, MDS-AML, and de novo AML. And I should say in particular MDS, because majority of our responders were within the MDS group. And as such, it may now serve as a bridge to hematopoietic stem cell transplantation. As I mentioned, there are, of course, now several possibilities to improve the current protocol and so on, but we are pretty optimistic. And in that, we are pretty optimistic that we may, in future trials, even get better results, but that, of course, remains to be seen. So with this, I'd just like to thank all our collaborators, in particular Andreas Björklund in the clinic. In the more recent years, he's been supported by Mattias Karlsten, Ebba Solberg in the lab, and these are all our collaborators, including Jeff Miller and Sarah Cooley from University of Minnesota, from which we have gotten quite a substantial amount of advice. And finally, Kalle Malmberg. And all of this was supported by the Swedish Cancer Society and the Tobias Foundation. And I should also acknowledge our nice collaboration that we have with the Nova Southeastern University. And with that, I'm done, Rich. Thank you.
Thank you. Thank you very much, Hans-Gustaf. That was a very stimulating, innovative presentation. And I'd like to ask everyone to please hold your questions till the end of all of the presentations, and then I'll ask all the speakers to come up and answer your questions. Our next speaker is a colleague at the NSU Cell Therapy Institute, who assistant professor, Dr. Beljanski, and he's going to talk on autophagy and stem cells, which has potentially also important implications for cancer therapy. So, Vlad.
Well, thank you very much. Everybody can hear me. Okay. So every time I talk about autophagy to increase people's interest in this pathway, I say one thing, which is if you manage to activate autophagy pathway frequently, you're going to live longer. Okay, now after this sentence, if you're not interested, then I don't know what to do. But this is a cartoon presentation of autophagy pathway, and it's really a cell on a diet. When there is not enough of nutrients and not enough of growth signals, specific sensors are activated, and they tell mTOR to stop protein synthesis. When mTOR stops signaling for protein synthesis, it activates cell recycling pathway, and that's what autophagy really is. It's a cell recycling pathway. It shares a lot of similarities with ubiquitin pathway. What happens is that cells take part of the ER membrane and then grow this structure called phagophore that when it's growing, it engulfs all kinds of cellular constituents. It can be organelles, smaller proteins, aggregates, and such. And then they form this vesicle called autophagosome. Well, autophagosome will then fuse with lysosome, and then when that happens, all these molecules and biomolecules that are delivered to lysosome are going to create small molecules that are then going to be used for cellular metabolism. So this is what cells do when they don't have enough of nutrients. And I encountered this pathway about 8 years ago because I was working on an anti-cancer drug that was killing cancer cells by activation of autophagy. So shown here is a cartoon of autophagy. And as I was reading about biology of this pathway, I came to realize it's important in so many diseases and has such a huge therapeutic implication and it's actually all unused right now. All these processes and all these diseases depend to at least a certain extent on autophagy. And 2 years ago, I started working on cell therapies, and then I realized that therapeutic cells must be starving at some point. They need to undergo some sort of cellular remodeling. They're probably exposed to the large extent to oxidative stress, definitely to immune response, and definitely they have some problems in metabolism. So autophagy must be really important for therapeutic cells, I thought. So we were very interested in immunological properties of stem cells and where autophagy stands with immunity. Well, it stands on two levels mostly. One is in antigen presentation, because some of the antigens are processed through autophagy as well before they're loaded into the MHCs. That's one big contribution. And the other one is secretion of cytokines, because autophagy secretes through canonical and non-canonical ways affects secretion of cytokines. So it affects cells, immune cells that are going to be interacting with cells that are going autophagy. It also affects cells, all cells that are secreting cytokines. So it must have an important role in the immune response. So we came with a hypothesis in which autophagy likely contributes to inflammation response of MSCs because it can regulate levels of inflammation-related genes and pathways. So to test this hypothesis, we're using Miami cells, and part of that because we live close to Miami, and part of that is because they are isolated, they are more primitive MSCs. So they are also isolated from bone marrow, and they can give rise to the large number of different differentiated cells. They are probably more preferred to work with compared to the embryonic stem cells, and definitely I was more interested in using them than classical MSCs because their potential for differentiation is a little bit higher. So we're using two drugs to either stimulate or block autophagy pathway. One is tamoxifen that blocks pro-survival molecules, and by doing that, it induces the pathway. And the other drug we're using is chloroquine. What chloroquine does, it acidifies lysosomes and then blocks fusion between autophagosomes and lysosomes. Therefore, it blocks actual recycling in the cell. Our experimental setup is we're treating Miami cells with these we're exposing them to these 6 different conditions. We're either treating them with single drugs or we add interferon gamma to see how these cells respond to inflammation. The day -1, we plate the cells, then we treat them as described 1 to 6, and then at day 4, we collect RNA and perform RNA sequencing. And data I'm going to be showing you came from the sequence 10 days ago, so it's fresh. So this is just to show you that there wasn't any toxicity during the treatments, and this is an assay that with a flow cytometry you can assess level of autophagosomes in the cell. Where it's vehicle, there is only normal level of autophagosomes. Also, interferon gamma doesn't really induce autophagy, but when you add tamoxifen, you see an increase in autophagic vesicles, and chloroquine, of course, because it blocks their degradation, so they accumulate in the cell. And then, of course, if you add interferon gamma to it, you kind of get the same thing. You're seeing an increase in autophagic vesicles. So this is the experimental setup, very simple. You take cells, you add interferon gamma, and then you add autophagy modulators. You put that into RNA sequencing, you process data, and then determine the pathway that responds to interferon gamma treatment in the presence of these drugs. So I'm going to show you some of the data here. It's been still all analyzed, but what I would like you to appreciate is the fact that when you add either chloroquine or tamoxifen, you're changing the expression level on the mRNA level of genes that belong, in this case, antigen presentation pathway. Just about every one of them has been changed. And I'm going to show you bar graphs to have a little more idea about the actual extent. Heat maps are just a very nice visual tool. Same thing with the Th1, Th2 activation pathway. You're seeing changes in gene expression when you add either chloroquine or tamoxifen. I'm going to show you actually chloroquine is much better a modulator here, or a modulator of the immune response, at least based on the molecular level. Interferon signaling pathway, again, we're seeing changes in the genes that belong to this pathway. Retinoid X receptor, liver X receptor, I put here because this is the pathway that kind of links metabolism and immune response. Again, there are changes in how the genes are responding to the treatments. And upstream analysis has shown that in spite of the fact that there is a change in gene expression, this is still inflammation, this is still a response that corresponds to upstream regulators of inflammation. So in either with chloroquine or with tamoxifen, this is inflammation response in these cells. Okay, so this is the actual values. When you add chloroquine, you are decreasing the levels of a number of HLAs here. In some case, like HLA-DOA, it's a 5-time increase. In some others, it's a little bit less. But in any case, for most of these molecules, you're seeing a decrease with chloroquine and to a lesser extent with tamoxifen. This is co-stimulatory molecules. With co-stimulatory molecules, we were surprised to find that chloroquine actually increases levels, for example, of IDO1 and IDO2, more efficient IDO2 than IDO1. It increases the levels of CD40, of HLA-G, all these molecules that are known to participate in immunoregulatory properties of stem cells. Interestingly enough, cytokines that are responding to inflammation response, usually with interferon gamma, as expected, for some of those, we're actually seeing an increase with chloroquine. And those cytokines play a role in the recruitment of T cells. So this is just a network to show you that when you treat cells with interferon, and then you co-treat with autophagy blocker or autophagy stimulator, your networks, and these networks contained within pathways and hundreds of genes, are changing. That means that biology of these cells is changing. And then we also had a chance to perform microRNA expression analysis. Just a reminder, microRNA is another level of regulation of gene expression. MicroRNAs bind to the mRNA and then send it for degradation, and this way they are decreasing their levels. So we assessed 800 most commonly studied microRNAs in a panel, and we're finding out when we look at the heat map microRNA versus the treatments, this is unsupervised clustering, we find out that the expression of these microRNAs is very much treatment specific, except for this here outlier with a tamoxifen treatment. Every one of them clustered in the same group based on the non-supervised clustering. So here are some of the microRNAs that are targeting antigen presentation pathway. And while we're still analyzing these data, I would like you to appreciate here is that co-treatment with chloroquine or tamoxifen regulates not only the levels of this microRNA, but also the types of microRNA that are being found in these samples. Therefore, this regulation is very complex and it happens on multiple levels. What I've put here is just as a reminder of what those microRNAs do. So this is antigen presentation pathway, and we put here some of the microRNAs that are also found in our data set that are regulating MHC2B complex, that are actually down-regulating MHC2B complex upon one of the treatments. So what I've shown you so far is that if you license your stem cells with interferon gamma treatment, you can change the way that they respond to interferon gamma treatment by co-treatment with either autophagy stimulator or autophagy autophagy blocker. What also is shown here is that chloroquine appears to be more efficient in decreasing antigen presentation pathway genes and in increasing co-stimulatory molecules. With microRNA analysis, we find out that the treatments with interferon gamma can further be affected by a selection of different microRNAs that are found in our set, both their quality and their quantity. And then we're arguing that if you incorporate stem cell therapy, or I think this could probably be valid for other therapies, cell therapies, that if you combine autophagy modulation with therapies, you can affect their therapeutic properties. What is it that is left to do? First of all, we have tons of data. We have to spend next several months to analyze all the data that came from this sequencing. For example, long non-coding RNA. Now NIH is giving grants that are only studying biological roles of long non-coding RNA because apparently they have a role, but nobody knows what they're doing. We have to look at the alternative gene splicing as well as the role of the pseudogenes, because for some of the microRNA data that I've shown you, we're seeing a decrease both in the gene expression and the microRNA expression, and we think that's because those microRNAs are now being further harvested with the pseudogenes, okay? Then all of this, of course, I'm a molecular guy, so all of this is very molecular. To really confirm this, we have to do first some co-culture assays to see if there is a biological effect of these stimulators and inhibitors. And of course, translational aspect will only be assessed properly when we use an animal model to see whether these treatments really help or hurt cell therapies. And this is just a picture of what we're going to be doing with studying of immunoregulatory, so ex vivo experiments in which we're going to be activating T cells in the presence of Miami cells to which autophagy has either been stimulated or inhibited. And it can be done genetically or it can be done pharmacologically. First, we're going to do pharmacologically because it's the easier thing to do. And then we're also going to be looking at the differentiation of T cells in the presence of Miami cells to which autophagy has been stimulated or inhibited. And with that, I'd like to thank first and foremost my partner, Lubov Nathanson, who's done all this great work in sorting out these RNA sequencing data from the sequencer. And I mean, she was a tremendous help here for the mathematical part of this project. Then Dr. Rossi and Hunter are from my lab at NSU. They did a lot of every day they're taking care of babysitting and changing diapers of these Miami cells because they're not easy to work with, and I thank them for that big time. Emily Blake did the actual RNA sequencing, And cells came from Dr. Paul Schiller and Dr. Gaetan Delacroix, who were at the time at the Miami VA. I think they're still there. So they lent us the cell to work with. Thank you very much for your attention.
Thank you, Vlad, for that very stimulating presentation. Our next speaker is Dr. Adil Duru. In fact, we became associated together through collaborations with Karolinska, where he trained prior to coming to the States, and we've developed very strong collaborations, and then he joined the Cell Therapy Institute as a colleague assistant professor recently. And the topic of his talk is novel immunotherapies for cancer, and I'll let Adil tell you the rest of the story. So, welcome. Okay. Forward. And there's your pointer. Okay, perfect.
Hello, thank you for the opportunity. I'd like to quickly start the presentation. It's a little bit cold here. So I'm an assistant professor at Southeastern University. Our overall aim is to develop novel cancer immunotherapies. I have a few complementing slides for Rich's introduction to NSU. So this is the most exciting triangle at the moment in my professional career, where we have the CCR, Center for Collaborative Research, and medical school just next to us, and then we are at NSU building a hospital together with HCA hospitals. So in this triangle, I am excited to have my lab and contribute to development of education, research, and clinical translation of what we do. A year ago, the previous picture you saw in the slide, it was me and Karin, my postdoc. Karin went back to Karolinska, but now we grow up, we have group members, external and internal collaborators, and also we incorporated students to our research. Our aim is to develop novel cancer immunotherapies at NSU. I will not repeat a lot about it, but we know one of the hallmarks of cancer is avoiding immune destruction. We know that now, last few days, we heard a lot about these things, so I won't repeat, but like we have most of the immune system infiltrated to the tumor, but the immunosuppressive tumor microenvironment somehow makes the tumor win the war. So very pioneering ideas last 15, 20 years came using cells like T cells, NK cell-based cancer immunotherapies, or other approaches using antibodies for cancer immunotherapies, which led to actually further development of these cells, reprogramming them, retargeting the tumor. So that led to an understanding that we need to do this. So, you heard a lot about last few days as well, and it's very popular. CAR T cell, CAR NK cell therapies recently, tumor-specific T cell receptor gene therapies also very popular recently, together with CAR T cells and NK cells. And they share something. What they share is like they try to make use of different components of immune systems to retarget and reprogram these cells and attack the tumor. Basically, like if we can give an example, chimeric antigen receptor, it combines the specificity of the antibody and the signaling components of killer cells and use an effector cell to rapidly target and eliminate the tumor cells. So what we have done, it's very promising. There are a few safety problems associated with them, but I'm 100% sure in certain cancer treatments they will be very important. And we also use the same approach from a different angle. And with Tolga, together, my colleague, he also did his PhD at Karolinska, we came up to combining different things. We wanted to combine natural killer cells with the specificity of the T cells by engineering and also getting target specificity and tumor specificity. So this is a project done mostly with Ayhan and Canan. Canan is my postdoctoral researcher. She's over there. Here we develop a novel approach and use NK cells for TCR gene therapy. It provides a novel source for cellular immunotherapy. It presents a final solution for TCR mispairing that is observed in T-cell receptor gene therapy. It actually also enables NK cells to target intracellularly processed antigens. And we applied for patent for this invention. This is together with Nova Southeastern University and Sabancı University. And I want to walk through a few slides to show how they work. So TCR expression on NK cell surface, with our technique, we can detect T-cell receptor with anti-TCR antibodies. We can also detect the T-cell receptor on the NK cell surface by using MHC class 1 pentamers. And then we can also observe easily TCR-mediated NK cell responses, which means actually antigen-specific MHC class 1 mediated responses when it comes to the degranulation. When the cells are fed with these externally with these peptides, we can see antigen-specific responses with the TCR, NK TCR cells, and actually the same cells can also recognize and respond to intracellularly processed antigens. In this case, these results are specific to tyrosinase antigen in complex with HLA-A2. And then we can also see both cytokine secretion when they're exposed to the antigen and also cytokine production. So this is a secretion of TNF-alpha, pro-inflammatory cytokines, and interferon gamma. And also we can see this is intracellular cytokine staining, so we can see also production of these cytokines upon antigen stimulation. But more importantly, are they killing? So NK TCR cells are killing the target cells in an antigen-specific manner like the T cells kill. These experiments were run with Acknowledgements platform. The blue line is normal responses to NK cells to the tumor cells. And then when we have the T-cell receptor, we see a very rapid response and a very rapid elimination of the tumor cells. And the next slide, I want to show it. This is the live cell imaging. Is it playing? I don't see from here. Yeah. So these are the antigen-negative melanoma cells. And on the bottom, we have the antigen-positive melanoma cells. And the red is the marker of death. So what we see is NK TCR cells, which is difficult here, I think, killing the target cells more efficiently than normal NK cells. But next, we wanted to see if these tumor cells, these NK cells can kill and target and kill the tumors in vivo. Dr. Benjosi and Dr. Anna Maria Yorgudaki helped to establish these experiments, and Dr. Yorgudaki, together with Canan, performed these experiments. And what we see is when we have the antigen-positive tumors and when we expose them to NK cells that carry the T-cell receptors specific to these antigens, we can see a significant reduction of the antigen-expressing tumor. So overall, what we want to do with this platform, with this technology, we would like to integrate this technology to what we are doing at NSU. Because at CCR we have several different core facilities led by Dr. Sara Tavakoli. She's around, yeah. She's also here. And also CCR will be a kind of a hub in this biomedical triangle that we will process the biological samples, try to develop cell therapy-based or exosome-based clinical products, and then we want to take it back to the hospital, like as a translational medicine pathway. So for this one, I am also the director of the cell therapy core facility, one of these core facilities. Together with Robin and Michelle and all other scientists, we are developing some pipelines for translational research pipelines at NSU. One of them is the application of the NK-TCR project for clinic. We collaborate with Dr. Tom Temple, and Canan is also leading this project. It's basically to make sarcoma cell lines, which is being done at cell therapy core facility, and also identify tumor-specific T-cell receptors to combine with the NK cells that we have. And with the recent news that I heard today from Hans-Gustaf, increase of MHC class 1 and responders might also give a little bit more better angle for this pipeline. And also, we are designing similar pipelines, trying to adapt these things and try to design our pipelines modular, so that we can adapt to different cases. So together with Anna Maria and Tom Temple, we are also trying to develop a CAR identification and optimization, or antibody identification, and then chimeric antigen receptor optimization platform. And that's it. Thank you very much for listening. This is a research of a lot of people, but I would like to thank our Karolinska collaborators, Hans-Gustaf, Adnan Ashur, Otti Hovatta, Evren Alıcı, whole group of Evren Alıcı, actually where I did my postdoctoral studies, Tom Temple for his support, and Jove for his leadership at Cell Therapy Institute and his persistence in keeping Karolinska collaboration alive, and also my close collaborators, Tolga and Evren and his groups. Thank you very much.
Thank you, Adil, for that very exciting presentation. Our next speaker is Dr. Wendy Weston. She comes to us actually from the University of Miami, UM. I used to be at University of Michigan, so whenever I see UM, I think Ann Arbor. But this is University of Miami here, where we are, at the tumor bank. So Dr. Weston was working with Dr. Tom Temple and colleagues from Vivex who were speaking earlier today, including Dr. Tim Gainey. And so we had a session earlier today on exosomes. So those of you who attended, you got a good introduction to that, and you'll hear more about it today. Wendy's going to talk about exosomes and cancer, and she's recently joined us at the Cell Therapy Institute. So, Wendy.
Thank you, Rich. Thank you, Rich. Good afternoon, everybody. Thank you for coming to see this talk. I am employed by Vivex Biomedical, which was formerly the UM Tissue Bank, and I work as a faculty at Nova Southeastern University, assistant professor. I'm going to talk about cancer and exosomes. Exosome formation occurs through endocytosis, formation of endosomes, maturation of that process, which forms these ILVs, or interluminal vesicles, inside of the multivesicular bodies. These multivesicular bodies will then fuse with the membrane and release their contents into the extracellular space. They've also been shown to be released by direct budding from the plasma membrane. There's a lot of controversy about the characterization of exosomes. The size of exosomes has been said to range from 50 to 100, from 30 to 200. It seems that the general consensus has been that they range from 30 to 150 nanometers. So they're very, very small. They're smaller than apoptotic bodies, and they're smaller than microvesicles for the most part. They contain a double lipid layer, and they enable intracellular communication without direct cell-cell contact. That's the key statement there. They have a round or cup-shaped morphology, and they express CD63, ALEX, CD9, and CD81 at the very least commonly. So what ends up in exosomes? Now, we hear that a lot. We've been hearing a lot of talk about exosomes, but a lot ends up in exosomes, and I think that gets overlooked quite a bit. We get a lot of proteins. These are cytosolic proteins. These are membrane proteins. That's the tetraspanins, the CD9s, 63s, and 81. The endosome membrane proteins, transport proteins, heat shock proteins, and these are the endosomal sorting complex, which is one of the sorting mechanisms used to sort cargo into exosomes. There are 3 sorting mechanisms known so far. This is one of them that's ubiquitin-based. There are 2 others. One is lipid-based. Lipids, of course. Exosomes have a tendency to have lipid raft components on them. DNA is incorporated into exosomes. Mostly it's found in apoptotic bodies, but DNA can be found in exosomes. It can be taken up by recipient tissues and cells, and that signal, that mutagenic signal, can be expressed, which can induce malignancy. Messenger RNA, long-known cutting RNAs, as we've already heard, and microRNAs, of course, as we all know at this point. The microRNAs can be incorporated and are functional in the recipient cells. They are mature microRNAs being transported extracellularly, which is pretty amazing. When it was first found, they were trying to figure out how on earth could that be, because microRNAs in the extracellular environment are rapidly degraded. And there's a specific, what they call an exomotif, that controls the loading of microRNAs specifically into exosomes. So what do these do? Well, they do everything that you've heard about today. They do antigen presentation, they do immune regulation, tissue development, remodeling, spread of infectious disease, epigenetic changes, and of course transcriptional communication. They're taken up by recipient cells in a variety of ways. This receptor-ligand interaction will result in intracellular signaling that will have an effect upon the recipient cell. You can get fusion, which will dump the contents directly into the recipient cell, which will transfer the RNA, DNA, proteins, and so on. You can also get endocytosis of the entire exosome. From this mechanism, you can both get intracellular receptor-mediated signaling mechanisms, as well as release of the cargo into the recipient cell. So in tumor cells, tumors secrete exosomes constitutively. They are continuously, well, tumor cells are continuously subjected to stressors. These stressors include hypoxia. Exosomes from hypoxic cells are different from exosomes from cells in normoxia. Same cells, different conditions, the exosomes are different. Acidic conditions, which is typical of a tumor environment. Acidic conditions will stimulate the release of exosomes and will enhance their cell fusion capabilities. Heat, as we talked about, you heard from Dr. Temple's presentation earlier, heat can be used to induce heat shock proteins, which will activate certain signaling mechanisms inside exosomes. Chemotherapeutic agents, irradiation, starvation, these are all things that we're aware of are stressors upon cells. They modulate the immune response against a tumor. They promote immunosuppression by inducing T cell apoptosis, by inducing dendritic cell differentiation and NK cell cytotoxicity, and by inducing immunosuppressive myeloid suppressor cells and regulatory T cells. They also promote tumor progression. They do this by communicating between the tumor and the stroma cells and activating proliferative and angiogenic pathways. And this is just a small portion of all of the things that exosomes are responsible for. But exosomes derived from tumors, this is a pretty wordy slide, so we'll try and make it easier. The cargo transported in TDEs, which is tumor-derived exosomes, reflects the cargo profile of the cancer cells. These tumor-derived exosomes can induce epithelial to mesenchymal transitions. They also enhance migratory activity, but exosomes in general can't do this. Tumor-derived exosomes influence invasion into blood vessels. They facilitate organotrophic metastasis by establishing a seed so that the metastasizing cancer cell can be attracted. So it's the soil and seed hypothesis. So if the seed and the host are compatible, and this is facilitated by exosomes, then the metastasizing cancer cell can travel there and engraft. Tumor-derived exosomes are sufficient to redirect this. So if an organ is not normally attracting the cancer metastasizing cell, tumor-derived exosomes can create that environment. Tumor-derived exosomes establish an immunosuppressive environment, as I already discussed. They use evasion of the immune system. They can inhibit the effector cells, stimulate regulatory T cells, and so on. Tumor-derived exosomes create a pro-tumorigenic inflammatory response. So that's an awful lot. And this is all the microRNAs found in tumor-derived exosomes that have been shown to play a significant role in cancer progression in various cancers. There is a much larger list available, but I didn't want to try to put that on a slide. So what are we doing? We're trying to detect and characterize cancer using the exosomes that are being secreted from those cancers. You can use urine, tumors, or blood to do this. You isolate the exosomes from those, and you try to see whether there is an increase in expression of exosomes, because exosomes are expressed about twice as much in tumor environments as they are normally. Then we take those same exosomes, and we will check and see if there are lipid surface markers, RNAs, so on and so forth, that are differentially expressed between healthy tissue and tumor tissue. And of course, from the tumor tissue, you see this earlier, whether it's malignant, metastatic, aggressive, or responsive. So this is my favorite slide. When you look at a cancer progression, you're looking at a single or multiple genetic mutation with maybe epigenetic changes, results in transformation. You get continued changes from that, resulting in malignancy, which will then progress to epithelial mesenchymal transition, which will then progress to vascularization, metastasis, seeding, and growth in the new tissue. Exosomes from every single one of these processes must undergo every single one of these processes, at least. So they must be sorted into exosomes in their original cell. They must be released from those cells. They must be taken up by the recipient tissues, and they must deliver their cargo to those tissues. Every one of these processes has varieties and sub-processes. And if we want to try and find out what each one of these contributes to a particular cancer, then we have to look at all of these pathways. That's a lot. But it can be done. And we're new in the field of exosomes, relatively speaking. And we're compiling data that will start to put all of these together. For every single cancer, these are going to be different. Not all of them, but there's going to be something significantly different between every single cancer with all of these pathways. That's what we need to find out. In order to do that, in particular, we're looking at sarcoma and melanoma. We're comparing the tumor and the serum. We're isolating the exosomes from that using a size exclusion chromatography method. Then we're looking at the size distribution of those exosomes and differential expression of proteins, lipids, RNAs, all the nucleic acids. And we're trying to relate those to the processes of sorting the tumor cargo into the exosomes, which is specific to cell types, the release of the exosomes from the cells, which is specific to release mechanisms, which is specific to cell types, the seeding of the recipient tissues, which is specific to tissue types, the uptake by the recipient tissues, which is specific to the original releasing cell type, The delivery of cargo to the recipient cells, again, original cell type, and the response by the recipient cells, which has to do with the cargo, of course. So with this, 5X has been funding this research in collaboration with NSU. Tracy Anderson has been the promoter of this intense research, and Dr. Timothy Gainey is the one who first mentioned exosomes to me and started me on the crazy path, for which I am eternally grateful. At NOVA, I would like to thank Dr. Temple for all of his help in providing samples and ideas. And Dr. Duru and Shannon Murray, I'm in collaboration with both the sarcoma, melanoma, multiple myeloma, and also lung cancer grants that we're attempting to gain currently. And Ida Hafstrand from Karolinska has been doing our size exclusion chromatography and is fabulous, so I would certainly like to mention her name and to say thank you even though she's not here. Thank you very much.
Thank you. Thank you for that. Okay. Thank you for that very informative talk, Dr. Weston. And now I'd like to ask all of the speakers to come up, and we'll have a panel discussion. I'd like to just kick off with a question myself, and then we'll open it up to the floor for additional questions. So my first, these live? Okay. My first question is for Dr. Hans-Gustaf Ljunggren, and in particular, in terms of the comparisons between NK-based cancer immunotherapy and T-cell-based immunotherapy, what are your views on the pluses and minuses of each approach? Because right now, I know this is a broad question, but T-cell immunotherapy is extremely hot out there. NK therapy could be the next type of cancer immunotherapy. So I think there is interest in knowing how you perceive, compare the two.
Yeah, that's not an easy question. I think T-cell immunotherapy has taken off before NK cell based immunotherapy, maybe in part because there are more people working with T-cells, more people have T-cells as their comfort zone. When it comes to questions like their cytotoxic ability, I would say it depends, of course, on exactly what type of cells you have, etc., but it's probably comparable. An advantage with NK cells as opposed to T cells is, of course, that the risk for graft versus host reactions is much smaller. Both type of cells are curable. You can put in whatever type of molecule you would like into the cells to direct their specificity. Both type of cells are scalable. You can produce huge numbers. T cells might be better equipped to penetrate some solid tumors, possibly. NK cells have a natural set of receptors for hematopoietic cells in particular, but can also target some solid tumors. These are some issues. I'm sure that there are many people here who have opinions on it. But
Thank you, Hans, because I know that was a tough question. But I think that what we're all beginning to see now is that even though CAR T has already taken off and we're seeing FDA approval and lots of clinical trials, that there's going to be a strong contender for cancer immunotherapy with natural killer cells. So I'd like to open the floor up for questions from the audience.
From your perspective, perspective, having a good handle on this process in a controlled research environment, what your opinion is of the importance of autophagy as we handle cell therapies in a clinical setting. Many of the cells are provided as cryopreserved cells, but in different media. When they're thawed, they're generally kept around. Sometimes procedures in the OR might take a while before the cells are administered, so they're in very low nutrient media. And my suspicion is that autophagy is a very common occurrence after cells are being prepared for clinical administration, and what impact that might have.
So thank you for this question. And actually, one of the reasons why I started doing this was because there is no data on what's happening with the cells when they are stressed out when it comes to autophagy. How does stressing out of therapeutic cells before they're injected or when they're injected, what their genetic makeup looks like? What happens with these cells? How does that affect their therapeutic properties? Does it increase or decrease? Unfortunately, I don't have the answer yet to the question whether it's a good thing or a bad thing, but only those functional assays and in vivo assays that I proposed will give that answer. However, based on the RNA sequencing data that I looked at, it looks like autophagy is a good thing for them to for the long-term survival in a patient, but maybe not so good when it comes to their immunomodulatory properties. That's I think, conclusion we can safely make based on the RNA-seq data.
So following up on that question, Dr. Beljanski, in terms of cancer immunotherapy, how can you see autophagy contributing to that therapeutic modality?
Yes, I think that's a very good question. And again, we don't know exactly with cancer immunotherapeutic cells how them being stressed, stressing them before they're injected into a patient, how does that change their ability to secrete granzyme or porphyrin, how that actually affects their long-term survival in a patient, because we know that those therapeutic cells are going to disappear after a relatively short period of time. So it means they're not very happy when they're injected. We know very little about that, and I'm trying to develop an ex vivo model to maybe get some answers to what's happening with them under stress conditions.
Questions from the audience? Yes, Katarina?
Very nice presentations, all of you. My question is for Hans-Gustaf. The change in ratio of HLA-E and class 1, is that a relative change because you actually have lysis of the, the changes in the ratios of the HLA-E and class 1 positive cells after infusion, is that just a difference in ratio? Do you actually have an upregulation of those molecules?
So it's a change in expression of HLA and HLA, total class 1 and HLA-E on the residual tumor cells. So you wonder if, can you repeat the question?
Well, my question is if the non-expressing cells are lysed, and that's why you see a relative increase, actually.
Yeah,
but
okay,
not an
so
increase
yeah,
in
so
absolute
the
numbers.
mechanism behind what we refer to as immunoediting, whether that is not, we don't know. It could be so that within the total tumor population there's a degree of heterogeneity, and those cells that are first or best lysed are the class 1 lower cells, and thus you have class 1 higher cells remaining. But it could also be so that the total inflammatory response that is elicited during the process of immunotherapy, resulting in heightened levels of interferon gamma, or so may also stimulate tumor cells to express high levels of class 1. We don't really know why.
Would it be beneficial to test cytotoxicity ex vivo prior to infusion and choosing the donor?
Yeah, it's a very good question. You know, we thought a lot about that before we initiated our trial, but I think what many people do in these early type of trials is that eventually you cannot think too much. Rather, you set the protocol, you go for it, and you spend quite a lot of efforts in then studying, examining the outcome, try to correlate the outcome with a number of parameters, etc. And but it's definitely tempting to try to identify patients that would have a large or a significant chance of responding, because it is quite a difficult therapy that we put them through. So we would like to have even more responders in the future, I would say. Physicians, of course, give us the most critical, the most difficult cases all the time. That's the challenge.
Okay. I have a question for Dr. Duru. The NK TCR strategy is very clever. These are NK cells genetically modified with T cell receptor. So how, in terms of the studies that you've done so far, I presume these are still NK cells, but what kind of T cell characteristics, if any, are you seeing with the T cell receptor genetic modification in these cells?
I mean, to give a little bit more introduction, natural killer cells already have several components of T cell signaling and effector molecules. So there are basically certain missing parts of it. So by basically building up this system, you can mediate T cell receptor responses on natural killer cells. But these cells, one of the answers is these cells still maintain their natural killer capacity, so they are still natural killer cells. They still have, for example, one of the things to check out, like the cell responses to K562 cells, it's not changing, but at the same time, when they have the T cell receptor and when they encounter their cognate peptide MHC complex on a target tumor cell, they go and kill like T cells. In general, it's a little bit linked to your first question to Hans-Gustaf as well. I think it's too late to distinguish between natural killer cells or T cells. I think what we will combine from these cells in one of the cells, it can be T cells or NK cells, what we will combine, which properties we learned from other immune cells, we merge them and retarget tumor. I think this is how I think. It's not like one favorite cell. It's like how you make use of the whole immune system in a one product.
Okay, great. Again, open up to the audience. Any questions out there? It's hard to see with the bright lights. Okay, we have a question right over here.
One question for Dr. Weston. It seems very interesting that you have the endocytosis, and so basically you get an insight out of what was a surface receptor becomes internalized to the exosome. How does this regulate the internal machinery back to the nucleus when you've changed kind of the ligand binding and what might be the activation sites, what would have been internally or now outside the exosome?
Well, it's really interesting that not only do some of these receptors get internalized within the exosomes, but they are also retained on the outside, which is something that's forgotten a lot of times, that although the membrane fusion for the exit of the exosomes from the cell, of course, is going to include receptors on the exosomes, they are originally born from endocytosis of the plasma membrane of the original cell. And so they do have receptors usually within lipid rafts from the original cell, which is one of the reasons that we can see that quite often and we can characterize from that. And of course, many receptors activate and have signaling regardless of whether they are embedded in the membrane or not. So if they are within the exosomes and they are then released to another cell, they can either be moved to the membrane of the recipient tissue cell, where they would have an activity, or they can act themselves, not all of receptors, of course, but some receptors can act without actually being embedded in a membrane and have an effect.
Okay. Well, with that, I'd like to thank all of our speakers for their very innovative, exciting, and stimulating talks. So please join me in thanking our speakers.